Eastern Indonesia time, and I believe it is 5 p.m. in Japanese Standard Time. So we will start today's guest lecture. The title is Human Cell Manufacturing for Clinical Applications. And this afternoon we are honored. We are very much honored to have a very well-known, famous professor from Osaka University, Professor Masahiro Kino-oka, or Kino-oka Sensei. I would like to introduce first his biography for the audience, for the students, lecturers that attend this guest lecture, so that we know a little bit more about our today's lecture. So, Professor Masahiro Kino-oka, Ph.D., is a professor in Department of Biotechnology, Osaka University. Kino-oka Sensei is one of leaders in the field of manufacturing for cell and tissue product. He is an individual who seems to be able to contribute to the standardization of the cell processing facility. So his current research interests are bioprocess engineering to establish the cell and tissue manufacturing system, including the fields of bioreaction engineering, bioprocess design, and stem cell engineering. So advocating cell manufacturability, which will lead to the stable and cost-saving system for manufacturing of stem cell. So Kino-oka Sensei earned his BS, Bachelor of Science in Department of Chemical Engineering, Osaka University in 1989, and Master of Science in 1991. Then he became research assistant at the Department of Chemical Engineering, Osaka University from 1991 to 2000, having the project bioreactor and process design for plant tissue culture. After getting the PhD in 1996, Kino-oka Sensei spent 1 year as a visiting staff in the Department of Chemical Engineering, National Institute of Switzerland, ETH, in Zurich, and started the new project for bioreactor design for human cells towards clinical application. Kino-oka Sensei returned to Japan in 1997. He was promoted twice in his 5-year tenure as assistant professor in Department of Chemical Engineering, Osaka University in 2000, and then as associate professor in the year 2003, also this department. And finally, he earned full professor position in 2009 in Department of Biotechnology, Osaka University. So prior to this Department of Biotechnology, Kino-oka Sensei had many experiences with bioreactor and process design for both plant and human cell cultures. Currently, he started new project for manufacturing, including the facility design of cell and tissue processing, development of cell manufacturing and processing system for industrialization of regenerative medicine by Japan Agency for Medical Research and Development, or AMED, for development of guidance for cells and tissue processing, good cell and tissue practice. Also, to develop the facility, he has joined the International Standard Organization to be international member in ISO TC 198 or WG9 for aseptic processing, as well as ISO TC 276 or WG4 for bioprocessing standard. In addition, Kino-oka Sensei becomes board of director for the Japanese Society for Regenerative Medicine, JSRM, and Health Science Council member in Ministry of Health, Labor, and Welfare, collaborating with the regulatory side of Japanese FDA, which is PMDA, to establish the guideline for aseptic cell processing. Kino-oka Sensei also established recently Kotozukuri Consortium for Cell Manufacturability since 2016 to develop practical system for cell manufacturing, guidance for regulation and standard, as well as human resources for the process design. Several companies gathered to open their laboratories inside Osaka University and making up the core of this consortium management. So, and before we start, Kino-oka Sensei, I would like to introduce first to our attendants here. So mostly are biomedical engineering students within Swiss German University. We are located in Tangerang, so nearby Jakarta, just the south of Jakarta. We have this small university, private university, and most of the students that attend this lecture is from the biomedical engineering department, and it will be of interest for them because they will be getting the exposure to stem cell and tissue engineering from you. And also we have several lecturers coming here, among others Dr. Muhammad Fatoni, also a lecturer in our biomedical engineering. Then Mr. Aulia Iskandar. There's also Ibu Dela Rahmawati, who is currently in Osaka University as well for her postgraduate study in doctoral degree. And and we have also some others, some alumni, and also guests. So we also open for registration for other from other participants from outside our university if they are interested to also listen to your lecture. I think without further ado, I would like to give the time and opportunity to you to give your STEAM lecture. Thank you.
Okay, thank you very much. Kind introduction, Daddy Sensei. And thank you very much for the invitation. It's my honor to be here to have a lecture for you. I'm Masahiro Kino-oka. Please call me that just Masa is enough for me. So my topic is human cell manufacturing for the clinical application. Especially I'm not a biologist. First, I have to mention, as Daddy Sensei mentioned, my background is chemical engineering, kind of the manufacturing technology I want to develop. I use the cells for the production of something. One of the application is a clinical application as a regenerative medicine product. That's why I want to contribute this field. So today I would like to mention the many matters step by step. If you have a question, of course you can interrupt me to have a presentation. Please tell me the question as much as possible. I want to reply. Okay, now I'm here. I'm also going to staff. So this is my laboratory member in one laboratory. I feel that too many students and too many staff in my laboratory because later I will mention I have many collaborations with companies person. So many companies person gather to develop the manufacturing process in my laboratory. Now is around 60 members. Student is 28 students and the others are staff. Okay, so that's the introduction. I want to skip this one. So the phrase Osaka, maybe I hope you understand Japan, of course, but you don't know the Osaka. Maybe you know the Tokyo and the Kyoto. So our university is close to Osaka. After, finishing the COVID-19, please come to Osaka because we have many, entertainment, especially for the food. Okay, so Osaka and also Japan, very easy to have a good trip. So after finishing the COVID-19, please come. Especially Osaka is well known for the merchant city. So the Tokyo area is a government. Kyoto area is traditional capital. And Osaka is a merchant city. So quite different colors between Tokyo, Kyoto, and Osaka. So first, I want to introduce our laboratory activity. I hope you will see the many video image because I often taking a video like this. So for example, I want to explain step by step. This is a mesenchymal stem cell culture. You will see a very unique shape of the cells during the culture because we develop the surface for the, intrinsic stimulation of the mesenchymal stem cells. So at the moment, I will not mention, but if you're interested, please contact me. And also here, this is iPS cell culture, induced pluripotent stem cells. I collaborate with many professors in Japan for the iPS cells. So especially I prepare the kinetic project for the kinetic analysis for the deviation of the undifferentiation in human iPSCs. So, and also I have, my background is not cell culture. My background is computational work. So I want to realize the computational culture surface in computer. I mean the in silico culture. So this is a kind of the iPS cell culture and the cornea sheet for, this is a culture for the cornea sheet. Cornea is the eye, surface of the eye. So we want to cultivate in computer, not practical computer. Because first I have to mention, I don't like culture. Okay, I like cells, but I don't like, very troublesome. I don't want to do experiment. So that's why I prepare the culture in computer. And here, video already stopped. I try to again. Okay, so this is a bioreactor system. And just a moment. Okay, slightly you see the cardiac muscle cells. Some beating occurs derived from iPS cells to cultivate prepare the cardiac muscle cells. We prepare the suspension culture system for using the bioreactor system. And here, today's main topic, automation system. And also you will see some migration of cells, red cells and green cells in three-dimensional structure tissue. We prepare the cell sheet for the clinical application, transplantation, and as a research, we prepare some analysis of the dynamic behavior of the cells in the structure. These are, typical representative work, but what is my research? Okay, I want to use the stem cells. For the stem cells, for the clinical application, final product, we need the large amount of the cells. So that's why we want to expand the stem cells to keep in the, for example, undifferentiation. Okay. And sometimes in case of the iPS cells, we want to prepare several types of the cells. For example, if we want to get the muscles, we don't want to get the bone. So that means that we want to regulate the direction of differentiation. So I mean, looks like the first, I want to regulate the first step for the differentiation from the undifferentiated state of the iPS cells. And after preparation of the precursor cells, or differentiated cells, we also want to get the cell again. So I mean, I want to prepare the bioreactor system for the differentiated cells. Sometimes we need some mature cells. So from the immature cell to the mature cell, we perform the culture. And sometimes we want to get a three-dimensional structure for the clinical application. And also sometimes we need the blood vessel. I mean, it looks like the functional construct for the clinical application. Okay, So what is my target? I want to say again. My target is not cells. Okay? My target is these arrows. I want to get the whole arrows from the materials to the final product. After getting the whole arrows, we can perform the manufacturing. Okay? So my target is not cells species. My target is many arrows because patient want to get the cells. So as an engineer, for each arrow we develop. So for this purpose, looks like a very broad, project, not a deeper project. So I mean the very narrow but broad project for the development of the culture system. So for this purpose, we have several groups. First, of course, we have to know the stem cells. What is the stem cells? What is the characteristic of the cells? And what is the system for the cell expansion? What is the functional 3D tissue? And also that's my original project, cell simulator group. So that means the in silico culture because I don't like the culture. My staff cultivate. I don't like to cultivate. That's the point. Okay. So, but first, maybe you understand my target. Target is this arrow. Looks like the kinetics from material to the final product. For the expansion, we have several bioreactor systems like this. Not only smaller scale for the suspension culture of the iPS cells, for example, but also the larger scale, 5-liter culture. And sometimes we need some different type of the, culture system, shaking culture system. Sometimes we need the medium exchange device. Because, compared to the other cultures, such as microorganisms, plant cells, yeast, or plant cells, or CHO cells, animal cells, they don't have any medium change. Only mammary cells, human cells, have to medium change. Okay, because development of the medium is not perfect. We have to medium change, we have to replace the medium every 2 days or sometimes every day, sometimes every half a day. Okay, I don't like the culture. So that's why I don't want the culture. So that means we need the automation system. And also that we have a larger scale, 20 liter in my laboratory. Too big, sometimes too expensive if we start a culture. Okay, so but this variety of the culture system is important for the practical production of the cells. Looks like the scaling up from the smaller to larger. So I put some review articles for today's topic. One keyword is cell manufacturability. Later I want to introduce. So if you're interested, please check this reference. That's open. And also for the cell culture technique, my colleague, Associate Professor Mihei Kim, prepares several articles for the culture technique. If you're interested, please check. Maybe my name is very unique. KINO-OKA, easy to pick up my name in the database of the literature. So first I want to introduce what is cell manufacturability. Only one word, we are in the department of the biotechnology. My background is biochemical engineering. So we have the cells. That's a fundamental consideration to develop the manufacturing process. We have to know the cell feature, cell characteristic, how the cell feel during the culture. We also have to know the robotic system to develop the process. Both are important. So I want to join this field as an engineer for the cell manufacturing for the regenerative medicine. Okay. So what is the definition of the cell manufacturability? I introduced this one. Maybe some of the students, I hope you know what is the definition of manufacturability. Manufacturability have a definition in the engineering field. So to save the cost, to make the robust process. Looks like this one. That's a manufacturability. So what is a cell manufacturability? I define the capability of cell manufacturing through the process by bridging the biological aspect and the process aspect, bridging to develop the manufacturing process. That is a system in my case. Okay, can you imagine? Not so difficult, but not so easy. For example, I want to show this one example. If we want to cultivate, or machine put the motion for the pipetting, for example, output from the motion. Okay, so that they have a unit for the flow rate. But the cells have some input to make the response to output. That is a cell response system. Okay, but we don't know. In case of the flow rate, they have a unit. Most of the case, cell feel the shear stress. That's the input. So output, unit for the output of the motion, unit for the input of the cells. If we have easy to convert the units from the output to input, but most of the case, we don't know the unit for the cell input. That is a problem, very difficult to understand the cell response. So looks like the methodology, not the methodology. Methodology means the kind of the determination of the units. So we have to know both of them, biological aspect and process aspect, to understand, to prepare the manufacturing process. That is a cell manufacturability. So what is the design for the cell manufacturability? Practically, manufacturing design of the cell-based product for the clinical application in such a way that they are easy to manufacture through the simple, safe, and efficient. Efficient means the cost saving process with stable product quality and secure to customer. In this case, customer is a patient, by considering transportation and preparation outside of the factory. Not only inside the factory for the manufacturing, but also the outside factory. That's important because cell quality easy to change just before the injection of the cells into the patient. I mean the dosing, during the dosing, quite changing the quality of the cells. So this is a definition for to the design for cell manufacturability. Based on this concept, we start to consider how to develop the manufacturing process. So first, we start to consider the first is a system as an engineer. In this case, I consider the system that's a processes from the input to output. That is a system. Okay. So to obtain the output, in this case, product, how to prepare the good product, stable robustness of the processing. I mean, to get the stable quality of the product. First, we have to consider what is the input. If the input making the variation, easy to fluctuate the output quality. So what is the input? From the company for the sales, transportation to come to the one company to produce a final product of the sales. So the materials input is one of the input is sales. Also the ancillary materials. Ancillary material is a kind of the medium or solution of the buffer solution or some other additional factors. And also we need the culture vessels. If the change in the culture vessels, we will get a different quality. So input, if the change in the quality, I mean the quality variation causing the fluctuation of the quality of the final product. This fluctuation, I mean the variance, easy to understand. And also we have to consider not only system, but also the environment out of the system. From the environment, we got a noise. Here, intrinsic noise, such as temperature, such as cleanness, or dust, or something like this. If we get the noise, we will get the fluctuation of the product of output. So this concept is much easier for everybody, I hope. Because it looks like the cable, electrical signal cable, if the input signal bad, output signal bad, that's normal. And during the signaling using the electrical cable, they need a shield to prevent the noise. But in our case, our cell production, we also have to consider the intrinsic disorder. You know, the cells, I like cell, but very mysterious. We don't know the cells. That means uncertainty. Cell quality easy to change, and also that we don't know the cell component. We cannot estimate the mole number of the molecular level. And also the cells that are living. Living means easy to change by themselves. So that looks like the time dependency. If they're changing the time, so cell quality start to change. Okay, because of the living. And the tardive, what's the meaning of the tardive? Slightly difficult. So tardive, after putting some stimulation, changing later, not quick at the same time. I mean, for example, after putting the new medium, cell feels something, and cell have a response after 1 day, sometimes after 1 week, sometimes after 1 month. So that's a tardive. Okay, they have, and also due to our technological limitation, we can detect later. That's a time delay. So during the process, we have to consider the whole of them. That means the cell easy to fluctuate by themselves. I mean the intrinsic disorder occurs. That's a process. And also if we prepare the processes, first process step, second step, third step, and we get the final product, this intrinsic disorder expand more and more and more to get the fluctuation of the final product quality. That's a concept of the new concept, because the normal manufacturing, we don't want to care the intrinsic disorder. But in our case, we have to care. And also if we change the process later, I mean the process flexibility, the quality of the final product change. Due to the process flexibility, both want to change the process from conventional to the new technique. The final product sometimes to become the bad quality. I mean, changing the quality. So that means sometimes difficult to change the process after fixing the whole of the process. In the factory, we have to consider this one. In the upstream, I mean the medium preparation, cell expansion, differentiation, something. And also the downstream, cell separation, purification, dispensing, packaging, freezing, like this. And also that we have to care not only inside the factory, but also outside of the factory, such as the transportation from the company to the hospital, and also the preparation in hospital. We have to care the outstream. That's my word, not technical term, still not technical term, but upstream, downstream, and outstream also important. Outer stream also possible. So during the transportation, if the changing the temperature, changing the quality of the cells, during the thawing of the cells and washing the cells, cell quality start to change just before dosing. So there are many considerations to change the quality of the cells. To realize a good world, ideal world for the patient, who is the stakeholder we have to first consider? Of course, first, citizen, such as a patient, have to know. Other academic society have to join. Regulator have to understand, and the government have to support us. So these stakeholders to perform the regenerative medicine. So again, I want to mention that, of course, patient have to know well. To support the patient, the supporters necessary for regenerative medicine, I mean the kind of the volunteer, public. And to create the regenerative medicine, I mean the researchers that are creators. And for the performance, for the clinical application, that means the conductors, I mean the medical doctors. And looks like this one is our research. Technical supporters of regenerative medicine, I mean the engineer also have to contribute to this field. And also we need some support from the regulator. Regulator have to understand to approve the final product for the clinical application. So at the same time, these 6 position person have to understand well. So with 6 position person, we need some growing up to realize the region medicine. So as an engineer, what is the most important patterns? So in our university, in the graduate school of engineering, I established the consortium, Kotozukuri Consortium. Kotozukuri is in Japanese. Looks like the social development consortium for cell manufacturability. So the reason why we want to establish the ecosystem of various tools, human resources, and the regulation to realize the cell manufacturing for the clinical application. So I mean the logistics, materials, automation system, instrument, facility, test device, and database data analyst, and the standard operation procedure development, education, international standard, and regulation, also the capitals, strategy, patents, they are important to realize. So I mean the kind of the manufacturing, rule booking, and the human resources. At the same time, manufacturing, rule booking, and the human resources gather at the same time to develop. So that's a Kotozukuri in Japanese. So in our laboratory, we want to perform the Kotozukuri by using the consortium. So that means that not only manufacturing, development of the manufacturing tools, but also the regulation develop, the regulatory science, and also the human resources development, the education. So this is a kind of the video. Can you hear this video sound?
Yes, we can hear.
Yeah. Okay, thank you. You can hear.
Research development to speed up industrialization. Professor Masahiro Kino-oka's research group at Osaka University is working on Kotozukuri, an industrial system that will connect individual technologies in order to develop a manufacturing system for regenerative medicine. We established our centralized R&D lab at Osaka University, where we are collaborating with several companies and institutes in order to develop production technology. There are many problems with manufacturing living cell and tissue products. In order to solve these problems, various technologies must be connected and applied. By connecting people, rules, and the things they create, we will be able to widen the possibilities of cell manufacturability. Currently, in the centralized lab, a prototype production system is in operation. FMP, a flexible modular platform, is being used to modulate each function with an isolator and can connect, remove, and install. FMP also allows modules to be shared while maintaining long-term asepticity, promising high quality and reasonably priced manufacturing. We are also working to make sure our interface technology acquires international standardization. With our connecting technology, our goal is to promote Kotozukuri and achieve full-scale industrialization for regenerative medicine.
Okay, thank you very much. So that's our laboratory activity. So until now, we developed several, group, consortium, and we developed the, automation system. And this automation system can understand the, characteristic of the cell during the manufacturing. From here, I like to mention the further important based on the cell manufacturability. So just before starting the topic, first we have to distinguish what is the features for the cell manufacturing. So maybe some of the students can understand the producers is a microorganism, produce peptide as a drug. For example, this is a conventional production using the cells. Okay, that's also cell production, cell manufacturing. So in this case, if we want to prepare the peptide as a drug, first we need the master cells. We use the same cells, same stock every time for the production. And sterile and the large lot exist to start the upstream and downstream to purify the final product peptide, and also the storage and the transportation and dose into the patient. So this is a conventional system for the manufacturing of the drug using the cells. How about the animal cell to produce a protein such as antibody? In case of the CHO cells, produce the antibody, at the time we have to care the less stability of the product quality due to the process fluctuation, easy to fluctuate, changing the stability of the product quality. That's why the FDA, many people mention the process is a product. If the process slightly changed, product would be changed. So main stream from the master cells to the dosing is the same. How about our case? We use the human cells and we produce the human cells. Of course, we have to consider this less stability of the product quality due to the process fluctuation. Additionally, as I mentioned, quality, cells. What is the quality? Cell quality, we don't know. So that means the quality uncertainty in the molecular level, less downstream tech, and long-term production, very long-term, manufacturing duration. Easy to cause the intrinsic disorder and easy to fluctuation of the product quality in outstream. That's in case of looks like the iPS cells. iPS cells have a master cells. That's a sterile materials and starting a large lot. But there are no downstream, very difficult to purify the final products, such as today, retina epithelial pigment cells, I want to show later. And storage, due to the frozen product, in hospital, we have to thaw in and we have to thaw the cells and washing in hospital, changing the quality just before transplantation. And how about the other case? Mesenchymal stem cells, such as from the bone marrows, even though the allogenic, not donor cells to the patient, the limitation of the growth exists. That means that no master cells for the production. Sometimes we have to change the starter cells. We have to change the donors. So that means that we have to change the quality of the materials. That's a problem. And in case of the autologous cells, such as skin of the patient, personal use. No lot, produce to start as soon as possible after preparation of the product. So one product, one batch for one patient. That's very difficult to produce because the fluctuation of the production scale, non-assurance of sterility for the starting materials. These are very unique in case of for the aseptic processing. And again, I want to introduce using the different schematic drawings. From the patient, that's an autograft for the transplantation, or allograft means that from the donors, harvest the cells, isolation the cells, seeding, that's a primary culture, and expansion culture exists. During the primary culture, not for the growth, looks like the accumulation because of the cells from in the body, and now is in culture. That means the out of body ex vivo system. So that means that we need the accumulation time. And for the after preparation of the many house cells, sometimes we inject to the patient directly or prepare the three-dimensional structure as during the tissue culture for the transplantation. This is looks like this circle is a kind of the process. There are many features again. One, utilization of living cells and the tissue under the aseptic environment. So that means that we cannot sterilize. So the unsterile raw materials and final products, that means that we have to keep the good aseptic environment to prevent the contamination. Again, and also the avoidance of the contamination. And of course, human error we have to avoid. And during the culture, most of the cell can adhere on the culture vessels. That means very difficult to mixing. Even mixed state of the cell in the culture space. That means locationally, cell exists in the high dense. Locally, cell didn't exist. So that means the heterogeneity in space occurs. And also from the human, so that means the non-uniformity in cell population. Oh, sorry. And during the culture, I don't know the student can understand the batch culture. Batch means seeding and growing and harvesting and seeding again. That's one batch. Serial means one first batch, second batch, third batch, fourth batch. That means serial batch cultures with less reproducibility. During the cultures, cell makes elder, become elder. So that means that we have to care the age of the cells. And also for the patient, in case of the autologous, so the patient needs a specific size of such as a skin. So that means that easy to change the fluctuation in production scale. Normally for the production, we always produce the same amount of the cells. But in case of the autograft, patient fix the requirement. Patient require the given scale every time. So that means the fluctuation in production scale. And also compared to the bacterial culture, not so large culture scale. Because of bacterial culture, for example, 1 cubic meter, exists, but in our case, less than 10 liter. Sometimes we need a multi-item production system for the patient, bone, skin, something like this. And complexity of the, culture technique and decision making, very difficult. We need an expert. Restriction of the sampling and sensing. Due to the, limitation of the number of cells, we cannot measure the gene or protein during the process. So from this feature, how to cultivate, I want to introduce. This is my old laboratory. We need a safety cabinet. We need an incubator. And also inside the incubator, we put the same cultures with dishes. And this is a tea flask. We need, for the collection, we need a centrifugation system. And of course, we have to observe using a microscope. That's tools for the cultures. First, I want to introduce how to cultivate. For example, today at the 5 o'clock, before leaving from the university, one student checking the cells because she is a very good person. She want to care the cells well. So before leaving, she want to check the cells, for example. So we found the many cells. What shall we do? Is that a good or bad? That's a bad. If the large number of cells, on Monday, it's impossible to use. So that's why this student start to prepare the passage, from one vessel to another vessel quickly, because she has to leave from now to go to the movie theater because of the Friday night. Normally you can laugh, okay, but unfortunately Zoom, very difficult to hearing the sound, okay. So quickly she start to having the house some protocol for the passage. First, she has to remove the cells. Before removing using the enzyme, she has to wash the surface of the culture, and she will put some enzyme solution. Please check. She used many plastic stuff, plastic vessel and plastic pipette. That's a single use, okay, disposable. So that means expensive. I don't like the culture. I don't want to waste time and waste the money. So she's a good student, but very expensive just for the preparation of the cells. For the main experiment, she has to do something, okay? And also she has to pay for the preparation of the cells. So how to reduce the cost and the time? That's a problem. After collecting the cells, she start to make sure that she start to collect the cells using the centrifugation. So using the centrifuge, looks like the washing machine in the laundry, collecting the cells. For the explanation of this video, very long time. Actually, this protocol is 1 hour. Okay, do you like culture? Maybe no. I don't like culture. I like cells. That's a first point, starting point of my research. So after collecting the cells, she removed the supernatant and she resuspend using the fresh medium for the seeding of the cells in the new vessels. Okay, so maybe too long to see the video image, so that I want to close this video image. but a very long time. Okay, that's the problem. I don't want to do. So what is the technique of the culture? I want to show the left side video image in current my laboratory, and right side is a practical processing area for the clinical application. So the gauning is quite different. Maybe you feel this is right side video image is a gauning. Okay, gauning normally twice. Okay, twice. This is the second gauning. Please guess if you take the pants. That's a upper wear, easy. But if you take the pants, very difficult. Don't touch the wall. Don't sit on the seat. And don't put the pants on the floor. So that means only younger person can wear. Unfortunately, I cannot enter into the right side room. Okay, that's a problem. And also this wear is a single use. If you check in the cells enter into this room, and after checking, he left from this room. At the time, I don't know your money, but in USA dollars, maybe the $70 pay to enter into this room. Can you understand? Very expensive. 2 persons enter. So that means $140 USA dollars pay only for the administration, entrance. That's why the house is expensive. Do you like the culture? Maybe no. I don't like the culture. Okay. So how to improve this situation? First, we start to consider what is the aseptic technique. Because our operation in the liquid handling, pipetting like this, and the solid handling, culture vessel handling, this liquid and solid handling combination makes the operation. Okay, during the operation, we have to care the aseptic technique. And how about the motion stability? One person, another person, different, we will get a different result. But we have to prompt. We have to do quickly. Okay, because this culture vessel is outside of the incubator. That means a low temperature, maybe the catching cold. Okay, so we have to care the environment. So that means the prompt operation is required. So what shall we do? Unstable motion exists during the culture operation, and development of machinery system for cell processing will make the stable operation and reduce intrinsic disorder. That's my hypothesis. Practically, during the pipetting like this, one person, first generation of the product expert, sucking the medium and pouring the medium is looks like this time for one dish. The quickly sucking and gently pouring the medium. If we mention to the other second generation operator, this guy do slowly. So we get a different time for the one operation. Please guess, we have many dishes during the practical production, such as 50 or more. So that means the total operation time quite different. So that causes a different quality of the cells. Even though the pipetting. So that's why later I want to introduce the automation system, because the machinery system can perform well and stability stability of the motion is very high. So before starting the somehow say consideration for the cells, I want to introduce what is the 2D culture and what is the 3D culture. 2D culture is on surface. Cell can adhere on the cells. That's a mesenchymal stem cells. And also the 2D culture with coated surface. For example, sometimes we have to coat the surface to grow more. And also the static culture of the 3D, the aggregate like this. That's our iPS cells and the suspension culture like this. The main technique for the culture. And from viewpoint of the cells, medium exposed to the medium and gas from the outside of the medium. Gas come, oxygen come into the medium, oxygen in medium come to the cells. And also the culture surface sometimes stimulate for the growth and differentiation to the cells. Okay, that's an environment for the cells. How about the cell behavior? Maybe some of the students feeling sleepy. So please see this one. This is an octopus. I have a quiz. This is an octopus in the sea. How to move the octopus? Maybe you know the octopus. In Japan, I like octopus. I often eat octopus. Especially Osaka, we have an octopus bowl, very famous, very good taste. I like octopus bowl. Okay. So how to move the octopus? Do you know? Swim? No. Dancing? Running? How do you think? Octopus start running. Can you see again? 2 leg. Running. How about this octopus? This octopus handing rice and running. You doubt. Okay, maybe you doubt. Please see. That's from science. No doubt. Okay. Okay, I want to say everybody knows the octopus, but we don't know the octopus. That's the point. We have to check the cells. Then we understand the cells. Maybe when the student was children, you know the octopus. But until now, you don't know how to move. That's a science. Okay, for the biotechnology, we don't know anything. So always we have to check by ourselves. How about the neural stem cells? Neural stem cells, the neuron, original of the neurons. We check the cells using a movie. They can migrate because the neural stem cells, well known, is the floating cells. But during the culture, thinking, they start to migrate without leg. Normally, they don't have any leg. So we were surprised. So we checking the legs by staining. They have in Japanese, that's a culture surface, 3-dimensionally. They have a leg on the bottom surface. Okay, I was so surprised when I saw this one. Okay, so everything new in this field. So we have to check by ourselves. Don't believe others. So the first we developed the house monitoring system by using the my house camera and the lens, not microscope. So the house in this case, mammary epithelial cells dancing like this. I was so surprised, not cell division, migrate and dancing like this. Again, I want to show. After seeding, cells start to migrate and some of cells to gather and making the dancing as a colony like this. Okay, what's the meaning of this one? I don't know, but they make the migration in the sheet. So I want to mention you have to check the cell migration and cell can adhere on the surface. We have to check the cell substrate adhesion. That's an integrin-based phenomena. And also the between cells, cell-cell connection also important to understand how the cadherin-based phenomena. So these migration, substrate, and cell-cell connection, this triangle relationship is important to understand the cells. How about the culture? After seeding, cell attach like this and elongate and start to elongate and also the making the cell division. And sometimes they can make the motion like this, rounding each as a pair. Okay, they have some meaning. If the migration more actively, that means the good cells. Okay, so motion is important to understand the cell healthy. So this is one of the example of the aggregate of the iPS cells. I will not mention in detail, but during the culture of the aggregate, one iPS cells, one type of the iPS cells, another type of the iPS cell, both are iPS cells, they start to change the phenomena. So it looks like this one. After putting this reagent, they can grow more. Some of this type of cells, they cannot grow anymore. They start to die. But if we add this lock inhibitor, they can survive. So both are iPS cells, but they have some characteristic each other. And so they have some property, even though the house aggregation. And we check some phenomena. During the aggregation, this green is the cells, three-dimensional cross-section image. Green is the dead cells. Red is the live cells. This is a top view. Again, I want to say, dead cell is from the inside of the aggregate to move to the outside of the aggregate. So this living cell push out the dead cells. Okay. Looks like the iPS cell want to gather between living cells. So dead cell want to throw away. Okay. During the culture. I feel just an interest point. We don't know this matter, so we found this one. And so to understand the phenomena, we try to prepare the machinery system just for the medium change or for the aggregate. Maybe you cannot see here is a small aggregate. This is a one vessel, only because this aggregate is around 50 micrometer in diameter. So the house is very small, so sometimes difficult to medium change. That's why I prepare the machinery system for the medium change. So we prepared two types of the flow, medium flow. Low looks like very slow medium change system, very fast medium change system using the iPS cells, this type and this type. For this D2 type, the growth by changing the medium flow is just looks like the same, but in this case, stick cells. You see the some positive position. I mean dotted. And during the time course, after medium change, they start to push some cells from inside to outside during the culture. So this is a kind of the good student medium change. This is a kind of the bad hand student medium change. If the good student can prepare the good spherical shape of the aggregate, but bad student getting the bad shape of the aggregate. Even though the medium, only medium change, we can get a different shape. This phenomena causing the different level of the differentiation later. So actually, we measure the several shape. So the x-axis is the shape from the round shape to the bad shape. Y-axis is the growing. So after medium change, first, if the good medium change, they can keep the spherical shape during the culture, but the bad medium change, first flux, a high flux, causing the bad shape like this. In Japanese. So that's due to the collagen, they produce a collagen type 1. So in case of the weak, this thick stock have a small amount of the collagen type 1. I mean the ECM formation, easy to fluctuate, collapse the partially by medium change, medium flow. And after collapse of the wall of the collagen, from the center, cells come to outside, push out. So looks like the eggs like this. Actually, we put some ECM, collagen type 1, as a red color. They have a good eggshell, looks like the eggshell, but they don't have any good eggshell like this, so easy to fluctuate the eggshell. Actually, this part, we stain with the LAC1. LAC1 is a kind of the indicator of the migration activity. After they come from the center to outer, they start to migrate more. So if we start the differentiation from this IPS, we will get a different IPS cell, different cells. Okay. Do I still have a time? It's okay?
Yes. Sensei, you still have time, yeah.
Okay, so after understanding this culture, I want to introduce machinery system. Maybe you can understand the technique is important. Some bad technique causing the different shape of the cells. So to develop the practical production of the cell production system for the cell manufacturing. So we apply the isolator system. Isolator system is looks like this one. So box style, chamber style of the system. Inside here, using the gloves, operation occurs under the good environment, I mean the aseptic processing. To introduce cells or materials from outside, we put the pass box here, and after decontamination cleanup, we can install into this area. And starting the culture, we can keep the cells in the incubator. For the passage, we have a centrifuge. Inside the isolated system in the operation area, we put looks like this one. Using this one, that's a very high robustness. They have a high robustness of the aseptic environment. This technique already done in the pharmaceutical production. So for the manufacturing, practically we put the 2 persons for the operation. One is one director. From left side operator to right side, they move to the dish and starting medium change like this. But we feel bad operation. She is a very high level of the expert, but sometimes the difficult operation, easy to vibrate due to the gloves and the bad pipetting system. So first shall we do? Machinery system is desired. So before starting the machinery system, we also propose a flexible modular platform. We separate the modules, the manual operation modules, various modules for the incubator or some others, and passbox module from the one system. And if we prepare the good interface between modules, same interface, we can connect and disconnect easily. Looks like the Lego block. If we put some good interface, we can create many systems. So that means that standardization of the interface, that's very important because many companies produce a different system, but how to connect each other, that's important. So, and we propose some facility to use isolated system. People can enter from here and gowning to enter the room. Okay, so that's a system for this is a room for the isolated system. So this is this regulation is under the ISO 18362. We developed by ourselves in the international standard. And in this room, we focus on the multi-purpose automation system, compatible modules, independent operation modules. Independent means incubator disconnect after finishing the one protocol to connect to the independent station to supply gas and the signal or air electricity. So to connect each other, interface technology developed, and much less people operator. That's much better. So that means few operator in this area. We thinking about 3 or 4 persons, including the one expert. So that's our idea. So we developed many instruments for each, supported by the Japanese government, by ourselves. And for the sampling of the menu of something like this, we also put some sampling device to measure something. Also the observation, as I mentioned, important. We prepare the incubator with microscope or observation system. So that's our laboratory system, collaborated with the company. So we prepare the automation system here, incubator modules, and the manual operation to enter in for the entrance of the materials. And between manual operation and automation, we prepare the transfer port between, and we prepare the interface for each module. That's a video image for the manufacture operation. Between modules, we bring to the different and to connect each other. That's a semi-automatic connection system. And after finishing one protocol for the incubator, we bring to the station to supply energy, gas, and exchange signals. And if necessary, we can connect to other, modules due to the same interface. And for the system like this, and for this is a kind of the prototype, but we can apply into the manufacturing, practical manufacturing, because we can validate all of them. So I often prepare the practical machine. But later I want to introduce this motion of the robot is good or bad. We have to care. So from viewpoint of the flexible modular platform, I want to introduce how to use. We put the materials in the material system to connect to the automation system. And also that we bring the cells and connect each other within 10 minutes. And starting the medium change, for example, that's around 1 hour. After finishing, this module detach to bring to others. And the other module connect and starting the different operation and disconnect and connect again. So for each operation around 1 hour, connection and disconnection is within 10 minutes. That means that in a daytime, for example, 10 batches possible to operate. So that means that we can prepare the parallel production of the cells. We prepare some incubator from the smaller tissue, larger number of the tissue, and dish observation system collaborate with other companies. And for the manual operation, we also can connect this one to the manual operation and automation system, of course, and with the software to count the cells. For the interface, we prepare the larger size, automation, smaller size, and manual installing system like this. So we can prepare the several type of the interface and compatible each other. So what is the operation? Which is better, manual operation or machinery operation? I prepare the several protocol for the machinery operation and manual operation. First, we consider the fine. For the automation system, we need a fine and predetermined arrangement of the plastic stuff within millimeter order. If the changing the different place, robotic arm cannot catch. So that means that we have to fix a good position before starting the operation. And also machinery system, simple and troublesome operator. Again, I want to say, I don't like the culture. The simple and troublesome operation, machinery system can perform well. But we also need a manual operation because we have an eye to check the position of the vessels, easy to catch. I mean the coarse and flexible arrangement of the plastic stuff. So we can perform. So I mean the manual operation sometimes advantage. And sometimes complicate and rapid operation necessary to by hand. So we need both of them. Okay, that's a point. Most of the researchers focus on only for the automation. So that's a nonsense. For the practical point, both are important. But we have to care how to connect between automated operation module and manual operation module. Again, we need a transfer system for the, connecting each other. That is the idea. So to develop the automation system, we have to put many devices the inside the operation room. So before starting, we prepare the virtual environment in computer. We prepare the same space in computer and practical machine. So first we check the machinery device position to consider the interfere. So after checking the position, we install the many devices practically. And also using this virtual software, virtual system, we prepare some motion checking in advance, and we install it into the practical machinery system. This is a kind of sampling operation from the one module to another module. And practical operation is the right side, like this. Just a moment. Okay. So at the time, didn't synchronize between, but we can synchronize practically. So we can change the acceleration and speed and something like in the computer, and we apply. So, but what is a good motion and bad motion? This idea to measure the motion, this idea is from my son. My son likes the TV game. You know, the Wii or PS4 or the PlayStation or something like this. So that if we move the remote controller inside the TV, they can move, synchronize, because we have a sensor for the gravity force. So if the shaking inside the computer feeling the gravity force to transfer into the speed of the motion in computer or in TV. Looks like the TV game. So we put the motion. I often say the motion calculator. We can put some gravity sensors here, a battery and the memory. And during the operation in in automation system with this system, we can measure the gravity force for XYZ axis, so that we can understand the acceleration mode and which motion is suitable, and feedback again. We compare the manual operation using this vessel with the automation system. This is a practical video image. Right side is manual operation for medium change. Left side is the automation system. So they have a same protocol for the medium change. Okay. So we checking the gravity force for XYZ axis. Right side operator is our laboratory expert, very good, excellent expert. So after measuring the operation motions, we compare right side. Maybe the small, but you cannot see the value, but y-axis is the gravity force for x, y, z axes. And maybe you see the many peaks, especially for the z-axis, high peaks exist. The maximum gravity force is 3.3 G. Small time, accidentally they have some higher gravity force. Unfortunately, first time, robotic system have a larger gravity force. The bad operation by robotic system. Maybe you know that manual operation having some motion, acceleration and deceleration, and looks like a smooth motion by hand. Okay, but robotic machine normally, from viewpoint of the simple program, acceleration, same speed and deceleration. That's all. no change of the acceleration deceleration during the operation. So that means a very simple program is not a good operation, but we can understand which motion is bad using the video image. So the improvement is possible. So actually this how the gravity force looks like the roller coaster in Japan. Roller's highest gravity force in the Asian Enka in the Fuji-Q highland close to the Tokyo area, close to the Fuji, Mount Fuji. That's a 3.67 G. So that means if the cell, I become the cell, I don't want to survive. Very sad. So the very high gravity force for the cells, I want to say. So we improve the motion by checking the video image with the motion calculator. And next step, I try to prepare some automation system for the practical retina and epithelial pigment cells. So this culture, IPS cells, expansion of the IPS cell culture, and starting the differentiation and maturation of the RP, I mean the retinal pigment epithelial cells, it takes 117 days. My student don't want to do. Okay, so just with the expert we perform. Okay, because maybe it takes time. Every 2 days or every day have to make a change. Maybe my student don't like the culture. Okay, I either don't like culture. So my expert can perform. So for the IPS cell culture like this, and during the culture for the differentiation into the retina pigment epithelial cells, looks like this one. Confluent, many cells, and looks like the fragile layered structure. If the bad motion shaking more, we will see that this type of the cell many. This part looks like the fragile part. So we need the good motion, gentle motion during the process. So the only expert can perform well. So we try to prepare the machinery system for this purpose. So where is the bad hand? We want to get the gold hand, the master's hand in the robotics system. What is a good hand? What is a bad hand? We can measure. So we want to skip. So we measure the motion. We improve the gravity, less gravity force, acceleration, G. So we perform the medium change, left stop, and tapping to harvest the cells for the passage, or washing using the PBS, or dispensing into the vials. So from the first, this part is a manual operation. That's a thawing process. Quickly we have to do. So that means we perform the manual operation. After seeding first time into the culture vessel, from here we perform only using the machinery system. So we prepare the robotic arm system. So how about the advantage of the machinery system? Our expert, unfortunately, we try to have 6 times. Only 1 time, my expert can perform. So that's very difficult. Okay, can you feel the difficulty? But machinery system, first trial, successful. Later, same motion means successful every time. But unfortunately, last time, I don't know the reason why, but it didn't. So that means that once after tune up the machinery system, same operation means causing the same result. The stability of the process is much higher in using the machinery system compared to the process using the manual operation. So from the first, we obtain the many cells. Practically, these cells only we need 10 to 5 cells. So once starting the production, we can prepare the more than 1,000 patient eyes transplantation. So that means we perform well during the long time. I don't like the culture, but I can perform with my friend. With my friend means with my robot. That's how the stability of the process. But we have to know the reason. So to understand the reason, we need several tools such as a motion calibrator or dispensing system to understand the change of the shape like this. We have to feel some features of the cells directly, and we can apply the machinery system. Okay, this is the final slide. Thank you for your attention. If you have a question, I'm very happy. Thank you.
Thank you, Kino-oka Sensei, for very interesting and intriguing presentation. I just knew now that motions during cell culture.
Yeah. Can you hear me? But I can hear you.
Yeah, maybe.
Yeah, we can connect each other.
So actually, there are several questions in the chat, Sensei.
Really?
Maybe we can start. We can start with the question.
Yeah.
So first is from Paholis. Paholis is one of our lecturer in Swiss German University, actually, Sensei. So the question is, how is the cell screening process to ensure that the cells are safety, free from any contaminants and other genetic abnormalities before culture?
Just a moment. Would you please explain again? Sorry.
How is the cell screening process to ensure that the cells are safety, free from any contamination and other genetics abnormality before culture?
Sterility. You want to say that? Okay,
the contamination.
Okay. For other materials, we have to check the sterility. We have to check the contamination check using the standard procedure for the sterility using the sterility test. Okay, so the same as the bacterial culture, we prepare the plate, special plate, not only for the aerobic bacteria but also the anaerobic bacteria plate. We paste the medium, okay, with the cells. And normally bacteria can float in the medium, and so after harvesting the medium, we put a special plate to check the colony of the bacteria. That's a simple way, but accuracy is not so high. That's a problem. And for the genetically stabilization, we sometimes have to check before shipment, especially for the iPS cells, easy to make the mutation. So that means during the process, at the end, we have to check sometimes. Okay, so not every time, but we have to care for the mutation. Is it your question? Is it okay?
So maybe I will ask Paholis. Is it
yes. Okay. Yeah. Thank you, Prof. Thank you, Mbak Dila. Thank you. Yeah. So thank you for the answer. Also, you answer the question. Yeah. So just sometimes because you might obtain the cells from certain people, right? Donor, right? So we have in my mind. So we have to make sure everything is okay for before the cell culture for example, HIV test or any other contaminations. Thank you, Prof. Kino. Yeah.
Okay. And for the autograft, or sometimes starting using for the starting of the culture, we use antibiotics to prevent the bacterial expansion. So and during the process, we try to move the antibiotics during the process. At the end, before starting the shipment of the product, we have to check the sterility again. So the starting, we can use antibiotics, but that's not perfect. So we have to remove the antibiotics later to check. Okay, sometimes if we add the antibiotics, bacteria start to make the spore. So that means the sleeping. So we cannot detect with antibiotics. So that means when we get the product, we have to wash without to remove the antibiotic, and we have to check the sterility test again. That's kind of the procedure. And also the virus, for the virus of the drug from the patient or donors, for the shipment from the hospital, they start to check, such as HIV or something. I forgot the name, was hepatitis, something like this. So we have to check the virus, to prevent distribution.
So regarding the antibiotics, well, we know that there are so many antibiotics, and antibiotics used depend on types of bacteria that might, So do you have any certain rules or standard? What kind of antibiotics?
We use some cocktail of the antibiotics, such as streptomycin and I forgot the
Kanamycin.
Gram-positive for gram-negative and mycoplasma like this. So it looks like the cocktails that we have a normal. But if you use a penicillin, we have to care for the patient.
Right.
Sometimes allergy occurs. So that means sometimes we have to care the patient. So sometimes we cannot use penicillin. Okay.
So in short, you are already calculate all about this from the very early on, right? Before culture, right? I mean, you have to consider everything.
Yes. Not only for the patient, but for the operator, we have to care. That means, not only patient. If the virus, for the operator first, we have to care. So that's other material attribute we have to check.
Okay. Thank you, Prof. Kino Sensei. Yeah.
Thank you.
Thank you, Mbak Dela.
Okay, maybe Pak Dedi is here. Not yet, I think. So maybe the second question. So Pak Dedi is here.
Yeah, so. Adela, you can continue because sometimes my internet is not stable, Kino-oka Sensei. I'm sorry.
Okay.
Yeah.
So the internet is not. Sorry. Okay. So another question is from maybe our alumni, San Diego Himawan. So thank you, Prof. Kino-oka, for your nice presentation. So he has two questions actually. One is in the downstream processing, what kind of purification method do you use? And the second question is how to overcome the metabolic burden in the manufactured cells. Thank you, Sensei.
Okay, first one is downstream you want to know. So for the cells clinical application, so practically we have a purification system such as a flow cytometry technique. The cell sorter system. But this speed, the preparation is not for fast. So that means normally for the downstream, first we have to prepare the separation process and then purification process. Both are important for the processing to prepare the final product. So, but we don't have any separation system and a strong purification system. So for the clinical cells for the clinical application. So that's a very big issue. So that's why that we have to prepare the good cells during the upstream, during the growing. We don't want to prepare the bad cells. Can you imagine? So but we have to check as a product, we have to check the quality of the cells. At the time, if the bad cells exist the specific level, more than specific level, we have to throw away all of them. I mean, the lot out, we often say. Whole product we have to throw away. Can you imagine? So we don't have any downstream and purification system. That means that if we cannot prepare the good cells well, we have to throw away the whole of the product at the same time. That means the failure, the big, impact of the failure for the cost. Can you imagine? If the purification, we can prepare the yield of the cells for the product. I mean the product number of the product change, but we can prepare the product. But in our case, downstream we don't have. So that means a lot out, throw away all of them. That's a problem. Okay, so no technique, for example. So how to develop the downstream? That's a point. Is it his first question? And how about the second question?
The second one is actually how to overcome the metabolic burden in manufactured cell. Maybe metabolic burden meaning contaminant. I don't know. San Diego, are you here?
Yeah, thank you, Professor, for your explanation. Yeah, what I mean is metabolic burden is the depletion of the energy from the organism itself. How to overcome this problem in your process system. Thank you.
Okay, so for the laboratory, every time we have to purchase from the same lot of the materials, and also from the same company. And for the practical production in company, for the preparation of the materials, I mean the reagent or media or something like this, that's a very important. They have they have to prepare the traceability to produce. So one company person for the cell manufacturing go to the material company to make the investment checking, okay, to get the traceability. So that means that we always have to get the same quality of the materials. That's a mission. Okay, the contaminant of a bacterial contamination is always from the cells or environment of the culture processing. So not from the materials. And the impurity of the materials also important. That's why the traceability of the materials is important. Is that your questions answer? Okay.
Yes, thank you, Professor.
Okay, sorry, still muted. So the next question, I think we have a lot of questions here. So Pak Deddy, maybe I will directly. Also, this is also from Pak Holis. Sensei, how many % of success rate in the cell implantation or injection? How many %?
For this process?
I think so, yeah.
For example, retina pigment epithelial cells derived from the iPS cells. Professor Masahiro Takahashi in Riken University, and now she becomes the president of the venture company, I forgot the name, a Vision Care company. So that she already have a clinical, clinical research for the 4 patient. Okay. So to check the safety, because we don't know we don't have any experience for the transplantation of the cells derived from the iPS cells. So in Japan, she is the first person, pioneer for the clinical application using the cells derived from the iPS cells. Now is 4 patient finish for the transplantation of the RP derived from the iPS cells. And the other case, okay, today I didn't mention exactly, but the cardiac muscle cells derived from the iPS cell. Professor Sawa in our university already done for the patient. 1 patient already done. So because the retina is not so many cells, just only 10 to 5 cells for 1 patient. So to prevent the risk of the mutation of the cells. But now in Japan, the move to the large amount of cell for the clinical application using the cardiac muscle cells derived from iPS cells, we already done. And also a cornea sheet, Professor Nishida in our university of optic surgery. So he already performed the cornea sheet derived from the iPS cell for the 1 patient. Using the iPS cells, we have of course, still less than 10 patients, not so many. But for the mesenchymal stem cells, okay, so I mean the bone marrow cells, JCR company already have approval for the clinical application for the GDHD, I forgot the full name, but for the many patients. In case of the skin from the patient, Japan Tissue Engineering, this company already done for the more than 1,000 patients. That's our patient cells. Cartilage, chondrocyte cells, this Japan Tissue Engineering company already get approval from the government and already done for the more than 100 patients. So we have many experiences, but our automation system is still not performed, still not installed into the company. Okay, so still we search the many collaborator into the company. But this machinery system we export to the Thailand, Bangkok University, KMUTT,
University of Technology.
Yeah, that's right, Tombuli. Yeah, we already export to the colleague for the future clinical application in the department of the chemical engineering. So so step by step we perform well.
Thank you, Sensei Kino.
Another 2 questions, Kino-oka Sensei, from our guest, Ibu Ratih. Can we use animal origin to culture the cell, such as FBS, for cell therapy?
Yeah. Okay. Yeah. So practically for the skin graft, I mean the epithelial sheet produced by the Japan Tissue Engineering. This manufacturing system is a very traditional system using the FBS. Okay, so already approved, not patient serum. I mean the fetal bovine serum they use, but this company person have to check the quality of the materials to go to the Australia because of the BPE bovine. I forgot the name of the disease. So but practically it's possible. But the washing process of the sheet is important. We have to check the residue of the FBS not detectable. That's a requirement from the government. Okay.
Another question is, what is the reagent that is being used for the passage cell? Is it using trypsin?
Okay, so trypsin material also important. So, but some of the company using trypsin, it's possible, and also the collagenase, and also the mixture of the enzyme. For the iPS cell culture, we use triple select. That's a mixture of the collagen and trypsin like this. So it's possible to apply. And the processing and this reagent processing covered by the GMP. So it's possible to apply. And also the chondrocyte cells, the Japan Tissue Engineering Company use collagen gels, aterocollagen gel derived from the bovine. But we can use this one. So if we purify or discuss with the regulator, we can use some of the reagent we can use. Okay. Of course, animal-free reagent is much better, of course, but sometimes impossible to replace.
Yeah. Thank you very much, Kino-oka Sensei. I think we will be closing the session soon. Before that, I would like to ask one question from education point of view. Would it be possible, for example, if one of our students would like to do internship or maybe final undergraduate thesis or project in your laboratory? Is there such possibility?
Yeah. For example, maybe there are some know well. So our department, we have many international students. So we have many lectures. Actually, we have many lectures in English. That's normal. So please discuss with her. And our laboratory, of course, we can accept some collaboration and exchange of the student. It's possible. Okay, so that's our pleasure. And practically in our laboratory, from Indonesia, now we have one student, and Malaysia is from 2 students, and Vietnam 1 student. Many Asian countries students exist, so very easy to survive in Japan. Actually, for maybe there are some will understand well. So we have good dishes for the Islam student.
Yes, in our faculty there are a lot of international students from Chinese also we have.
Thank you very much. I think that will be all for today's guest lecture. On behalf of our department, Biomedical Engineering, on behalf of our faculty and also university, Swiss German University, and also all of our students and staffs and also audiences, I would like to express our gratitude for Kino-oka Sensei for your time, your opportunity to share your experience in the cell manufacturability. This is something new also for me. And of course, we hope that we could expand this collaboration further. Thank you very much, Kino-oka Sensei, and see you in the future when the pandemic already gone. Thank you.
Thank
you very much, Kino Sensei. Thank you very much.
So
thank you very much.
Thank you, Sensei.
Thank you very much for the
important talk. Thank you, Mbak Dela.
Hi.
Ready? Yeah.
Okay. I would end the session for all. Thank you and see you and stay healthy.
Ada foto belum Mbak Dedi? Ada foto tuh.